The time slot between 1:15 and 2 will be used for hackathon teaming each day for the rest of the week. So you have a couple more opportunities to present still. If you would like to present, please do email me just to let me know you're interested so we can make sure everybody has enough time. Okay, so Bharat says you can present. So in the chat, while I'm going through these slides, if anybody who has an idea who has not presented yet wants to present, maybe we can give them time. I think there's value in letting people represent, but just as another reminder, this is also being live streamed. We have links here to the hackathon folders. I'm going to skip through some of this because I think people have seen it for the most part. For anybody who has not seen the hackathon information yet, this is in module 2.E in the Google Drive with the link that I posted a minute ago. If you have any questions about it, please email me and we can go over it. So this is just a link to some papers that we think are useful or would be good candidates for replication in, sorry, I'm just pasting the link here. That we think would be good candidates for replication in CompuCell3D. So if you are looking for inspiration, some of these papers may be a good starting point. Here's that link. Some important links here, which I believe I also have, but I'll post these in one big batch. So if you do have an idea, please post it to the shared teaming document. Put together a quick lightning talk presentation using the template in the folder and upload that to the folder. And like I said, notify me that you would like to present and we'll get you added to the schedule. So yes, some people, I think everybody who is here has presented before. So we've got James, Bharat, Max, and Liliana. So maybe what let's do is just start with James and we'll go through in the order that people posted in the chat. Today, so if you've got your lightning talk already, we need to cut them down a little bit for time. So I'm going to give people 3 minutes. And unfortunately, I'll have to be a little bit mercenary about cutting you off at 3 minutes, but please today, if you would just go over your there's 3 slides, your biological background, your modeling goals, and your potential teammates that you're looking for. We can go ahead and get at least a quick summary of your projects floated out in front of the whole group. So James, if you want to go ahead and take over the share.
Thanks, Hayden. I'll just get the screenshot. Okay, can everyone see the presentation?
Yep.
Okay, perfect. So I'm here today to briefly pitch you the idea that I've got, which is to try and replicate a within-host model of tuberculosis infection. So with regards to the background, tuberculosis is an infectious disease that's caused by the bacterium Mycobacterium tuberculosis. Typically, in 9 in every 10 cases, the infection is controlled. A load of immune cells are recruited and they aggregate and wall in the bacteria and contain it inside the structure known as a granuloma, an idea of which you can see in the diagram on the slide here. Unfortunately, in about 1 in every 10 cases, the bacteria is able to overwhelm this immune response and spread throughout the body, or in particular throughout the lungs, and causes active TB, which is important because in about 5 years from the initial infection, 50% of people with active TB will die if left untreated. These days, typically you'll get a load of antibiotics to try and get on top of this, and they'll normally be administered over the course of at least half a year or so. So it's a long regimen. And what can also happen is that even if you have an initial latent infection, it can reactivate later on, where the bacteria can break back out the granuloma, typically in immunocompromised individuals. And even if successfully treated with antibiotics, there is a possibility of relapse. So there is a model that I will be looking, or our team will be looking to replicate. So we would compare the outputs of our model to the existing agent-based model that has been developed to look into this, try and figure out what causes the different outcomes of latent TB versus active TB, there's a few hypotheses from the original paper, which suggests that where the initial inoculum is relative to any surrounding blood vessels is important, and the type of bacteria left at the end will tend to be less metabolically active. So we'll look into that, and if time, try and extend it and add in a few different cytokines, for example, or different types of immune cells. So as far as I'm concerned, I'm a first-year maths PhD student at the University of Bath in the UK. I've had a fair crash course in a lot of the biology on this in the last year or so, but it's fair to say I'm not really an expert just yet. And I have had some prior experience in Python, and the training has been very helpful, but again, I wouldn't claim any expertise here. So if anyone has any strong immunology or infectious disease background in biology, or consider themselves experts in Python, I'd be very happy to hear from you. In general, if you sound interested in this project, then yeah, that'd be great as well. At this point, I believe we have 3 in our team. I think Jacob's here today, and Vasileus, I'm not sure if he is or not. So possibly another 1 or maybe 2 teammates would be ideal, I think. Hoping to perhaps get a paper out of this and use this towards my PhD thesis. So any help will be greatly appreciated. So thanks for your time and your attention.
Great. Thank you, James. That was almost perfectly 3 minutes. So unfortunately, we don't have time for questions today. We will make sure that for the rest of the week, this hackathon time doesn't get overrun, so that we have plenty of time for other people to present. So let's see, who was second? Again, if you have any interest in participating in any of the projects that you see here today, go ahead and send each person an email. Their information should be in that shared document. So I think Bharat was next. Bharat, if you'd like to go ahead and do your 3-slide rundown.
Why don't speakers,
no.
sorry, just before we do that, why don't speakers put their emails also in the chat so that people have it there?
Yeah, so Bharat, you can go ahead.
Sure. So you can see the slides, right?
Yep, looks good.
Okay, great. So hello everyone. I'm an undergrad at Indian Institute of Science, Bangalore, and my project is going to be developing a wide-scale model to understanding colonization of cancer in the peritoneum, specifically ovarian cancer. So ovarian cancer spreads through a very non-conventional way, wherein it does not require blood. It spreads through peritoneal fluid. So it originates in the ovarian surface epithelium, and then the single cells which are exfoliated, which are more mesenchymal in nature, due to the motion of peritoneal fluid ends up at the mesothelium, which is here, the yellow and the green monolayer. And upon adhesion with receptors, these cancer cells release matrix metalloproteases, and also due to the pressure and division, they basically breach the basement membrane to reach the underneath collagen-rich matrix, where they form the metastatic lesion. Now, what I have done here, so the question of interest here is basically, if we just divide this problem and look at that monolayer which I was talking about, a single cell layer, which is made up of mesothelial cells, and if we age them, do we observe that there is a higher invasion, or the cancer cells are able to proliferate faster within those monolayers? So in this figure, that's my experimental project. In this figure on the left, you have those mesothelial cells in red. First, a confluent monolayer of it is made, and then green colored or GFP labeled cancer cells were added on top of them. And this was incubated for around 3 days. And what we see is when your mesothelial cells, which is shown on the left, are non-senescent, basically they're not aged by any means, we see some green patches which form within these monolayers, which are the cancer cells. However, when you make these mesothelial cells senescent using several methods like chemical treatment or gamma rays, you observe that the cancer cells are able to proliferate better, and they're also somehow clearing the mesothelial cells. You barely see any red cells here.
Brad, you have 1 minute left.
And that was quantitative. Wonderful. That was quantitatively done. It was seen that the area actually decreases and then cells are able to proliferate better within. And time-lapse images shows that this, if you focus on this area, these are how the cancer cells are able to kill the senescent cells. And they also causes some sort of lifting off from the substratum. Well, so the modeling goals is basically to use contact energy, growth, death, and chemical field plugins, which most of us are already aware of by now, and to see if the senescent and non-senescent cells are able to form any patches or some sort of pattern. Then basically quantifying by adding growth rate to it, making the cancer cells grow and cause some cytotoxic effect in the surrounding to see if and why senescent cells are somehow helping in the proliferation. And then the 3D model would be basically having cancer cells on top. And since we know that senescent cells secretes a lot of chemokines, these cancer cells get attracted to senescent cells and then doing clearance. Well, that's all. As of now, I have 2 teammates, but our team is open to more teammates who have any modeling experience and somewhat cancer biology. So, yeah, that's it.
Okay.
I'll stop sharing.
Thank you, Brad. Sorry, that was not, the sigh of exasperation was not at you. It was at what Javid just posted in the chat. The link to the old hackathon document keeps sneaking back into my shares, so I apologize for that. I will send the correct one now. Please do not add your information to the old one. Max, if you want to go ahead and queue your share up while I do that, we can get to yours in just a second. Thank you. Is it Javid?
Am I good to go? Or
Yeah, just give me 1 second here. Okay, so the correct teaming document link has been posted. Please use that one to update your information. I need to figure out why that old link keeps sneaking back in.
all right.
Okay.
Hi everyone, I'm Max Baga. I'm an undergraduate at Emory University. I'm going to keep this really brief because I've already presented and I actually have to run to catch a flight, but I'm here to present my potential model, influenza A pandemic strain evolution from co-infection and reassortment in humans. So quick, brief biological background. I've done influenza research in the past, and what I really like to study and look at is how pandemic strains evolve, and essentially the processes that 2 distinct strains will infect the same host and actually infect the same cell, and they'll exchange genetic information, as you can see on the diagram in the right, and produce sort of these recombinant viruses. And this practice is called reassortment and is what I want to specifically model. And essentially through reassortment of avian influenza strains, human strains, and even pig strains, you can evolve these influenza pandemics. I've done research in mammalian model hosts, avian host, and swine host, but I've never had the opportunity to test it in humans because obviously I can't go on and affect human beings and explore the results, but now I can do it through modeling. And that's what I hope to use CompuCell3D to do so. So simple modeling goals. 1, does reassortment in humans contribute to pandemic strain evolution, or is it mainly from other hosts? How does it behave locally? Does it evolve more in certain tissues? And is the viral fitness in humans increased through reassortment, or is the immune system presenting some sort of stringent, purifying selection? And I outlined sort of the project model here, but for the sake of time, I'll skip through this slide. A lot of the resource I'm reusing is my old work that I can use to sort of parameterize the model. I've done work quantifying reassortment, and I've done a lot of PBPK modeling in other areas, and I hope to draw on those skills to the model. And potential teammates, I've had a few people reach out about wanting to work with me, and I'm willing to work with anybody. I am new to CC3D myself. I'm very experienced with Python, but no modeling experience or influenza experience as well as required. And if you guys want to reach out, my email is max.baga@emory.edu. I put that in the chat, and I look forward to working with y'all.